All right, it's going.
It is?
Yeah. Might want to scoot in a little, John.
Yeah, everybody just scrunch in. There we go.
Is that good right there?
Looks great.
Can you see everyone?
Oh, yeah.
All right, get in here.
Everyone but you, Skylar. I'm going to put this up a little bit so we can see a little less stable and a little more face. Oh, yeah.
Okay.
So now we can just discuss all of that. Okay. Right.
So we decided to do it in a group because we're all here on campus. So we're just going to video us discussing the material.
All right. Do you have yours up?
I have it right here.
All right. So this is just going over the group problems.
Oh, just if we have any questions, right?
Yeah.
That which we did and handed in yesterday.
Mm-hmm.
Yeah.
Okay.
And then we have to answer those like 5 questions, right?
Mhm. Okay. So with the definitions, see, I didn't have any questions really on those. I guess I could have come up with a better definition for the chromosome set. I just said that we get one set of 23 inherited from dad, another set of 23 from mom.
Yeah, that's what I
That's
pretty much
basically
said
what
too.
it is.
And then, just to clarify, a homologous chromosome is one from mom and one from dad.
They're just similar.
They're like the same size.
Relatively
and
same
everything.
size. They're going to basically code for the same things.
But then the
Just have
sister
variances
chromatid is
in
like the exact copy that happens after meiosis.
Yeah. So what I get is like, so at DNA replication, you just have the one long strand of, it's like 2 separate chromatids
Yeah,
or something like that. It's connected to the one. And they call that like a diploid
Okay.
chromosome or
Uh-huh.
something. And then you have another one from mom. Is
Mhm.
that it? And then those 2 connect or something? Or do we have, like it's talking about, like a lot of the pictures and stuff like that I found online showed them separating 2, there's 2 little cross chromosomes, like the double
Yeah.
chromosome set being pulled apart, but there's one up here, one down
Mhm.
here. Are each of those their own, like one is from mom, one is from dad? Or is like 1/2 from mom, 1/2 from dad? That's what I had questions on with it.
Yeah. It's kind of what 102 is. It the 2 that are both from one, or is that the 2 separate ones coming?
Mhm.
Or is it one with its similar sister chromosome? Because that seems kind of.
So,
But yeah,
So-
everything else, I mean, made a lot of sense. Like I remember all the other stuff. Homozygous, heterozygous, dominant, recessive, all that. Did you get all that, John?
Yeah.
Okay.
okay, this shows homologous chromosomes. Oh, okay. So this shows these 2 are homologous or locus. Well, let's see. What is that?
Locus is the site of an allele.
Locus. Yeah, that's the
specific
side of the allele. These are homologous and these are homologous.
Mhm.
So when they connect together, they're a
Okay.
homologous pair right here. The sister chromatids are right here, and
Okay.
then there's some others down here.
Where are the sister chromatids?
These little ends right here, those are chromatids.
But they're the sister ones?
Yeah, these are sister chromatids when they're exactly identical like that. And then these are another set of sister chromatids.
So are these then, is this one homologous pair? And they each have one sister chromatid? Or is
Each
it,
of these is its own homologous.
so the blue is a homologous pair?
Yeah, because this
Oh,
is not homologous.
Click right here. This is explaining it good, I think. So like these are homologous chromosomes. These 4 here. See how it's saying that right there? And then this is
Oh.
a sister chromatid. These 2
Or a
are
copy
sister
of
chromatids
this?
because they're a copy of that.
Okay, so this is one homologous pair. These are the sister chromatids.
Right.
Oh, this is okay. So this is what they look like after they've replicated.
Yeah.
Okay.
So yeah, this is one
Okay.
pair or
That'll
whatever.
make more sense with the numbering then.
Yeah.
Okay, because I was confused on the numbering. And
And these are homologous chromosomes, but it says they're
non
non-sister because it's
those are on that
of
one and
each.
that's
yeah,
on that.
those are not sisters because they are like a copy, but they're not exactly a perfect copy, from what I understand.
Okay.
And
Yeah,
these
so-
two are non-homologous to each other.
Right.
Yes.
Okay.
Yeah, because they're not.
So one of these will be from mom, one will be from dad on these, right?
Yeah.
So-
Yeah.
And then ambivalent is when you get like two, but they're homologous, I guess, and they join together. Is that right?
Could be.
Ambivalent?
I don't remember what
Ambivalent.
ambivalent is.
Remember ambivalent?
It was in the
tetrads?
reading.
Yeah.
I just
I
don't
was just
remember.
wanting
Yeah.
to clarify with you guys because they like when you draw it,
Uh-huh.
it wants to know like, well, the synaptominal complex to me, and I'm
gotcha.
trying to get the difference. Like synaptominal complex is between one homologous pair, but ambivalent is when you get two or something like that together, like a
Yeah.
sister and the parent.
And the parent.
Is that right?
I think so.
Mhm.
Okay, that was something I
Yeah.
don't really.
Can you hand me my backpack? I got my pictures in there that we kind of compare.
Pictures are great.
Mhm.
Look at that.
Come on,
Well, I don't know
I
why.
think those were.
I decided to do that, but I was like, wait, I'm running out of room. So
Oh, yeah, I had to. I just
I
did a Word
confused.
document and they're like,
those
"What are you
look great.
doing?"
Yeah. Okay, so, and I only did my labeling on the parts when they first
Oh,
appeared.
okay.
Yeah, I didn't know if I
That
should
wasn't
continue or
yeah,
not.
that
I don't think
important.
so, so.
I got, you can see how I got lazier and lazier with my drawing. It went on.
I didn't get lazier and
But-
lazier. I kept
Yeah, I
going.
mean, no, I didn't.
I'm just kidding.
Mine are perfect.
I forgot we're being videoed.
I mean, I didn't really have any questions about these.
Yeah, the pictures. You can,
Yeah,
that was pretty straightforward.
Yeah.
We were like in the reading.
yeah. I was just curious.
Okay, so number 4 is where I hardcore struggled. I really do not
Oh,
understand that.
I forgot to do that one.
Oh. Dang.
John.
Okay. So let's seriously talk about those ones because now that we know what a chromosome is just one half of those little X's right there, basically. I think that's what it's asking. Then a homologous pair is two sets of those. That's weird.
But that
So
also
it
be a
says-
Why
set,
would they
then,
do
wouldn't
that?
it?
That's a set, yeah.
But it says the total number of chromosomes. And then
It's 48, so that means
48.
there's 24 sets.
And is that what it's asking with the pairs? So it asks for, the total number of chromosomes, the number of homologous chromosome pairs, and the number of sister chromatids present in each
yeah.
cell. So for the first one, prior to DNA replication, it just has the 48 diploid chromosomes
Right.
right there.
Yeah.
Meaning there's
That's what
going to
I
be
wrote.
24, because I looked up what a diploid chromosome is, and it's just,
They haven't,
it's two of
formed
them
yet.
together.
but they don't have a centromere yet.
Yeah, they don't have the centromere.
That's
put
what it's
in.
called, isn't it?
Yeah, the
Tremere is
centromere.
what connects.
Yeah.
Because once they have a centromere, it's called a tetrad.
Okay.
And then a haploid chromosome is just one
One of them.
individual double-stranded.
It's
Okay.
going to go
Or-
to a haploid cell or a gamete, so the end of meiosis too.
Mm-hmm.
Yeah. And then each of them has two sister chromatids. Each chromosome has two sister chromatids.
That so confuses me a little bit, the sister chromatids now, because I thought when we looked up the picture,
Yeah.
So confusing. Uh,
because to me it would make sense for the DNA replication to create another copy of a tetrad or whatever, right?
Mm-hmm.
Because then it's going to end up as two separate cells by the end of meiosis one.
Well,
Right?
you get something like these. You get the single-stranded things. and then DNA replication happens, and
So
well,
then,
let's see.
or do you get 1 and then it goes to, or no, 2 goes to 4?
Before, okay, there was a picture I saw on there. Let me look at it real quick. So, what was it? It was meiosis. Okay, this thing right here is on
That video.
YouTube video too. So this is all before DNA replication. This is basically DNA replication right here. So you have just single-stranded haploid ones. They
Make a sister
Duplicate.
chromatid, right?
make a sister chromatid, yeah.
Okay.
And that's what I put in my definition too with the sister chromatids is 2 identical copies formed by a single chromosome replicating. Both copies are joined together at a centromere. Each is 1/2 of a replicated chromosome. So,
Okay. Oh, yeah, because then it connects at the centromere.
Yep.
Okay.
all right. So, chromosomes. Pretty sure it's the 48, right?
That's what I have.
Homologous pairs,
24.
24.
That's what I wrote.
And then sister chromatids, I put 48 as well on that one.
Wouldn't that be 24 sister chromatids? Because if you had 24 originals,
Uh-huh.
We have 24 pairs of homologous chromosomes.
it's
Each of the chromosomes in that pair has a sister chromatid. So 24 times 2
That's
is
that's
the
where I
48.
got
48.
That's where we were
Yeah.
getting that.
okay.
And so,
because you remember it said like the homologous were like whatever these are terrible, but like these are homologous
Okay,
and
because
these are sister,
I'm just thinking
right? That's what that
Mhm.
picture.
homologous just means they're similar. It doesn't mean they're correlated. Just
Yeah.
similar. But when you see them connected at the centromere, that means there's a sister.
Yeah.
Yeah.
Okay, because I was just picturing the
Yeah.
tetrad then. Okay. So now we just got, like that one picture, that beginning one, we just got 48 of those guys floating around.
Mr. Artist. So yeah, I think it is 48 sister chromatids before replication.
Yeah. Now it's the hard stuff. But this is it. So homologous. Okay. So after meiosis is the next one. Is that right?
Beginning of
Beginning
metaphase
of
1.
metaphase 1. That's right. Yeah, not meiosis. That's the whole thing.
So at the beginning of metaphase 1, the I don't know even how to talk about this. The chromosome pairs are lining up at the metaphase plate,
Yes.
and the microtubules are connecting to the chromosome to the centromeres and about to pull them apart.
Mhm.
So I didn't think anything had changed.
So I put that DNA replication, it's going to double the number of chromosomes completely.
Right.
Because this is before that even happens.
Uh-huh.
And I looked at the whole so you have to I had to look at it in the whole perspective of it all. Because you get 96 individual chromosomes. That's what one half of these is. That's a chromosome.
Okay.
Right there. So there's now 96 of those. All in all, there's 48 homologous pairs. No, okay. So these all, oh gosh.
A diploid chromosome is like a complete set.
Yeah.
Because it's asking how many.
If it's a set, that means there's 2.
Yeah.
There was a website I saw this is the reason I did that. Because on the website it shows that there's going to be if in like some assays it talks about in DNA replication. Because we have 20
That's where sister
Three
chromatids
pairs.
come in.
Yeah, we have
Is that
the
S phase?
yeah the sister chromatids.
Uh-huh.
Technically, that's two sister chromatids is two chromosomes put together basically into one pair right there. But let's see. But it was talking about how like we have the 46 chromosomes total, 23 from mom, 23 from dad. But then after replication, we have 90. What was it like? It doubles.
92.
92. Yeah, we get 92 chromosomes. And that's just the individual leaves
Count. Yeah.
because they form into those. And then that makes up the 48 or 46 that get separated during meiosis.
It's basically like after the end of meiosis 2, there's 4 haploid cells. I just have 1 chromosome, but otherwise for us it'd be 23. And then 23 from the other parent. And then when they
22,
combine, or yeah.
Because isn't the sex separate or is it all in one?
Well, let's look at that through.
Oh, I think it is.
So we
Yeah.
have
Okay. Do you guys mind if we split like episodes?
No, we're
We
gonna
don't mind
we're
her
just gonna be
talking.
talking,
Yeah.
but if you don't mind, then
Okay,
yeah.
no, you're good.
Okay.
We just have to have a meeting too.
Yeah, not a problem.
Nothing to do.
Yeah, that's fine.
all right. So we have 23 of those. So with this example, there's going to be 24 of these, 24 of these, red and blue,
For 40 days.
So 48.
to get 48. So let's look at that real quick. So before DNA replication, that's what they have, right? So 48,
Yeah.
there's 24 homologous pairs.
Right.
Homologous pairs are one is from mom, one is from dad. They match each other kind of. Okay. And then the sister chromatids, there's 1, 2, and do we go 3, 4?
Oh,
Do each of those have, so there's 1, 2, 3, 4 sister chromatids within each homologous pair.
yeah, that's a good question because
I
if
think
they
this
all
one long
replicate,
thing is one. Is that what you're saying?
No, each of those, yeah, I'm not sure. I think that's right.
Yeah, I don't think this
'Cause I never
is 2.
saw-
I think this is 1. This is, it just looks separate because there's this
well,
thing,
that's
but
the
really,
centromere that connects.
Yeah,
and
but
it's
they're really
two
just
being
pinched.
connected by
Yeah.
one.
Okay.
yeah, so
that's
it's,
fine then.
so there's 4 in this thing right here.
For
But each chromosome has 2 sister chromatids. So we've got that first one right. DNA replication, that's right. Now, beginning of metaphase 1,
Nothing's changed. has
say
it?
there's,
well, they
Well,
become bivalent.
replication
Does that
would've-
change what they're how you count them? Because then they're going to connect. Like this is going to connect to that one, the red and the blue.
so look at this too. So this is another thing I put in that kind of solidified me thinking that that was 96 now and 48 and then 96. Because once you get to fertilization, fertilization is basically they're doubling again to give them that original 48 chromosomes,
Right.
They're just joining together.
joining back to, yeah. So in telophase, once I went through it, I had 24 chromosomes here, 12 pairs, 24 sister chromatids. It like separate, there's going to be half as many in this as there are in this one, between the two telophases.
Yeah, because this one's going to split into 2 where there'll be like a set or whatever.
Yeah.
But then that set will separate to their synchros for the haploid cells. So
Yeah.
it's like 1 for each cell. One's 1 half.
Mhm. Now, before that, before this telophase, we had the metaphase. Metaphase,
But just
we've got, that's just lining them up.
I didn't think-
Telophase is them separating, so you're going to have half as many in this telophase.
Yeah.
If we were to do that whole pattern, after fertilization, we'd only have 24 chromosomes before DNA replication.
But isn't it going to be just 24 at the end of telophase 2 for each cell?
Yeah,
Yeah.
at
that's
the
what
end
you're
of
saying,
telophase,
right?
yeah, at the end
Okay.
of telophase, you get 24.
Yeah.
But if we did not have,
That-
after DNA replication, if they didn't double into the 96 chromosomes,
Oh, because then you could split it up into 4 different.
yeah,
okay, I
we'd
see where
do
you're-
more splitting
yeah.
and we'd only have like 12 chromosomes
Because-
and then 6 homologous pairs. That
After the
doesn't-
96, it's going to split them into 2 cells each with 48, and those 2 cells are going to split into 2 for 24.
And then they double after fertilization. They
Yeah.
double. Does that make sense?
Yeah.
Do you think
Because
that's
they're
right?
fertilized.
I think it's right. I don't
Yeah.
know how else it would work.
Because this is I don't know why they'd put the prior to DNA replication and why it would be the same number of chromosomes
Yeah.
before and after DNA replication. Because it's going to double it.
Replicate.
Yeah, that makes sense.
Because I think, isn't there something happening before prophase?
Yeah, there's like an anaphase and what
No,
is it? No,
there
what
is.
it?
That's
Not
like,
anaphase. It's
there's going to be some sort of, that's
like
where the
the
sister
interphases.
chromatids come
Yeah.
in. There's a couple of different phases, I think.
Like
the one
S,
it mentions
isn't it like
is
an
the
S phase?
phase,
Yeah.
there's
and that's
an
where the
S
sister
phase,
chromatids form.
interphase
Okay.
S. That's what.
So we're saying there's 96 at the beginning of metaphase.
Yep.
48 haploid or homologous chromosome pairs, and 96 sister chromatids.
Yes.
Right?
That's what I thought. Yeah.
Okay.
Right. So 96, 48, 96.
And at the end of telophase, it drops down to 48.
Yep, 48, and then 24 homologous pairs.
And
then
48. And then at the end of telophase 2,
It'll be
it's down to
24.
24, 12, and 24.
And
That's
then
what I had.
after fertilization, it doubles,
It doubles
back
back
again.
up. That's the one I didn't really know.
That one I'm pretty sure. I looked at so many websites to figure that out. And then my mind keeps switching between whether it's the one-stranded chromosome or the double-stranded
Yeah, that's where I
chromosome.
was
And that's where I'm
getting
getting
drawing.
so confused with. And I keep thinking back to each one. So
Does that make
that's
sense
where
to
I'm
you, John?
frustrated.
How do you explain it? Okay. All right, let's move on to the stuff we know.
Okay.
Punnett Square.
Punnett
I love
You've
Punnett
been
Squares.
mentioned.
I like it when
Since freshman year, I
Since
remember in
Sunday,
high
yeah.
school, I remember doing.
Yeah.
Yeah.
Not the hard ones, but the easy
Yeah.
ones like this. So number 5, I had a 1-to-1 ratio. There's going to be two whatever they're called, heterozygous, and then
Too tall, too short.
the ratio of 1-to-1 ratio of heterozygous
Correct
tall
term is
plants and
dwarf
dwarf plants.
John. They don't like being called short. It's dwarf plants.
You're gonna
Is the
offend
proper way to
someone.
write it 1-to-1? I just wrote
I
1/2.
put 1/2 to 1/2, like 1/2 transfers.
Yeah, I mean,
Because I mean, that's not
Yeah,
a
I've seen. Yeah.
set in stone deal, but chances. I thought it was kind of a probability thing.
Yeah.
I don't think. We'll see, I guess, if she cares. She marks us off.
Don't pick me off.
Right.
I'm just writing. I don't know how
Just
you
kidding.
want us to write ratios.
We don't have to resend this thing. No.
Never mind.
Oh, okay.
Not
Yeah,
gonna
we're not
send it,
resending.
But hey,
Okay, number 6. So I don't know if we're supposed to. I did the whole thing, like the giant thing.
I did too. Yeah. The big,
Okay, all right. I think there's a better way to do it, but that's
we're,
okay.
we're supposed to learn how to do these. Like that was part of the little
right.
thing that they gave us is learn how to do multivalent
Yeah.
ones. I think that's the word for it. Is that it? But
So let's see. Did you guys get 3/8 smooth and yellow?
Yep.
mhm.
1/8 smooth and green?
I got 1/6.
I got 3/8 smooth yellow, 3/16 smooth
Mhm.
green, 3/8 constricted yellow, and then
Yes.
1/6 constricted green.
So why?
What did I do wrong?
How'd you
does
get
your
the
Punnett square
3/16
look?
smooth green?
1/16 smooth. Oh yeah.
Because there's the same amount of smooth green as there's constricted green. Right?
Yeah, 3/8 and 3/8.
3/8. I got 1/6.
So let's look at
No,
your
because you
comments.
have constricted green 1/16 and smooth green 3/16. Is that what you're saying, John?
Oh. So
Yeah,
yeah, I got
except I think these are like 2/16 because then I put it down to once or no something. Hold on.
this is okay.
Should
So
we go
we've
over the
got-
Punnett square?
Let's do
Yeah.
the Punnett square. real quick.
Okay.
So how's it set up? We got one. Okay, you made a cross between that has constricted pods, which is the negative. That's the recessive, right?
Constricted is
That's a
recessive. yes.
double small
Small
s. Small.
Yeah,
Yeah.
2 small S's.
Yeah.
Okay. With a heterozygous.
For seed color. So big Y, little y.
Okay. So where do I have that? I've got. So constricted, and then yellow.
So it's like this for the first one, right? 2 little S's, 1 big Y, and 1 little y.
Yes.
Let me see if I did.
And then for the second plant, it's heterozygous for both pod texture and seed color. So it's a big S and a little S and a big Y and a little Y. Right?
So all of these are little S's up top then on your.
They are for me.
Yeah,
Are they for you?
I didn't have it. Like that.
Yes.
That's weird. What happened? My autocorrect is changing some stuff too.
Oh.
So, and then I've got a big SY, big S, little y.
So the way I did it, let me just see if I did it right. I took this S and I put it with each of the 4 Ys. No. What I did was I took this S, this first S here, I did it with these 2 Ys for the second plant. It doesn't really matter either way. And then I took the other S and I did it with the second 2 Ys. And then I took the big S here, like you did here, and I put that one with a big Y and a little y, and then a big, and then the little S's with a big Y and a little y.
Yeah, it's
So
kind of
the
like
way-
this. So
The way you have it set up, that's exactly how I have it
Okay.
set up.
Let me make sure I've got this right here. So the first little Punnett square, you're gonna have a big S, little s, big Y, a big Y.
Where are you
Second
going
square,
down?
I'm going across.
Okay.
So the next one is a big S, big Y, little y.
Is this what you have too, John, so far?
Yeah, I think I screwed up on the second one down. I think I put a big Y where I was supposed to have a little Y.
So yeah, it helps that it's all S's and Y's, which look the same, both capital and lowercase.
exactly.
Good point.
Big S, big Y, big Y.
Mm-hmm.
Big S, big Y, little y.
Yep.
Big S, big Y, little y.
Yep.
Big S, little y, little y.
What if we just
I feel like,
counted,
yeah, let's just, okay, we
like,
got it, guys.
instead of counting the numbers,
So
the letters,
stupid.
let's count
All
how
right,
many,
there are
how many smooth greens we have.
Okay, let me recount them then. Smooth, and so that's the big, have a big S and a big Y.
I've got 6.
Yeah, I think that's what it should be. I think I have 7 now. And then 9 smooth green.
1, 2, 3,
6.
5, 6.
Smooth green?
Smooth and yellow.
Smooth and yellow.
Okay,
Yeah.
6 out of 16.
So
Okay.
smooth green would be
6
1.
out of 16, so 3/8 is what that is.
Big S.
And then let's go smooth green. So big, it's got to have a big
2.
S, and then
2 little
just
ones.
2 little Y's. So 1,
2.
and 2. Yep, I've got 2
Yep.
of those.
Okay.
So where'd I
Constricted
move
and
it?
yellow.
Wait a minute. Oh, that's where I moved this up.
6. So there's 6 constricted and yellow.
Constricted
So that
and
means
yellow.
there's 2 constricted and green.
4,
Wait, no.
6.
Constricted and 1.
Yep.
2.
1, 2. Okay, mine was right.
Okay.
Okay, wait. Okay, 6.
Again, 1/6.
Do you have, okay, so you've got the
I
3
don't even know how you simplified down to 1 6.
That's what I mean. I think I screwed up. I think. I
'Cause
must have been thinking
maybe you were
something
trying to
else,
write
'cause it
1/8.
might have to be 1/8. Yeah.
Yeah.
Autocorrect is messing with me.
That's the only way for it to make sense. So 3/8, 3/8, 1/8.
Yeah.
So 3/8 for smooth and yellow. 1/8 for smooth and green. 3/8 for constricted and yellow. 1/8 for constricted and green. Correct?
Yes.
Okay.
Okay, I think we can move on.
Whew. That was rough. And John, you did the giant Punnett square for this next one,
No, I did 4 little Punnett
like
squares.
the 128.
Yeah.
Imagine, okay, guys, do you have a big A, little a, small a, small a?
Yeah. Through every one of them. Oh, it's not.
No, I just did 4 little ones that got
And multiplied.
like half there, 1/4, 1/4, and I multiplied.
Okay.
And that got 1/64.
Same here.
Perfect.
Perfect. All right.
All right. Well, that's done.
That's done.
Now to start the actual assignment.
Good. I've got an hour before I gotta be in class. No, yes.
You got how long?
Biostats at 3:15. No,
Okay.
class is 4 times a week.
Wow.
Cool. Did you take it online?
Biostats?
Yeah.
No. Well,
We have to
No.
meet in class 2 days and then meet as groups 2 days.
Who you
But that's in
taking it from?
class. Promar.
that's who I took it from. I actually like that class though. Like he makes it
Doesn't seem bad
pretty.
so far. I heard he was good with the take.
He's really good. Like he's an easy A, I think. In that class. Okay.
Guys, we started. It's brave enough to come in.
I know.
That's right. You just had to
We're inspiring others for bravery.
Okay.
Okay.
So
Applying genetics from this just tells the instructions.
I think if you click that link, it'll-
Are you under group collaboration?
Because I can't. I love check marks. So
I do too. Especially with this new app.
um,
did you already download? Oh, there.
there it is.
Okay. Do you think we just have to do one turn in, or each of us turn in?
I think we'll each turn it in.
Okay.
That group work on it as a group, from what I understand.
Okay.
So this group collaboration thing, that's this
So should
hangout thing, right?
we just work on?
We're on
email
Conroe.
meters or something?
Yeah, since we're using
Yeah.
the laptop. Um. 375. Okay. So, suppose you perform the following cross. And it's a five letter one. Assume all these genes undergo independent assortment.
Good.
Okay.
Easy.
And then like, go to my notes.
When it says independent assortment.
Means they don't influence each other.
Oh.
So it means like, because there's some genes that would influence another gene, but these, yeah, it's just saying like A doesn't matter about what A,
Yeah.
like A's outcome doesn't depend on what B
Okay.
does or whatever. Because aren't there some that like it'll turn on or off a gene or something like that,
Yeah.
depending on what they.
So now we just make
Is this even asking what the Punnett square results are? What are the questions? What proportion? So yeah,
What portion of is expected to have that genotype?
that genotype.
So we need to do
So we
the-
do four. Oh, that's easy. Okay,
Yeah.
let's all do a Punnett square.
We all doing all of
Doing
them,
A's,
or
but
do
that's-
we want to split it up? I mean, it wouldn't hurt, I guess, to get practice.
Yeah.
They're not going to take one.
Wow. So you had to go to the amateur.
Wow, John. Sorry.
On camera too.
Yeah. You guys better not become best friends and not hang out. Okay. Deal?
Let's see.
Yeah. Yeah.
Why do they always choose letters that look the same?
I don't know.
I know. What?
Do T's and
Pick a Q or
A's.
something.
T's good.
Yeah, D is good. Oh,
Yeah, that's
What
right.
mean?
I'm already done with the bun square.
With a Punnett square.
just one.
Okay.
God.
All right, I'm done. Just kidding.
Yeah, I'm not gonna. I can guesstimate what all those.
I'm done.
Over here.
Well, me too. So
No, really? It made us roll. They're all a bunch of cheating people.
Oh, I haven't written anything out about them.
Oh, I'm writing all my info then. I should have
Oh.
told you when I'm
I
finished.
just finished the
totally
actual
would-
square.
I would have won.
actually, I'm calculating that.
Wow.
Oh, well,
Wait, did you read the questions? Oh, okay.
way ahead of you.
Wow.
Groups work better in competition.
What proportion of the offspring is expected to have that?
0.0029065. 0625, excuse me. So 1/64 times 0.2. How do I turn it into fraction? Hmm.
You got it's 3/4 for the big C, little C, right?
Big C little C.
No, it's one
It's
for
1/2.
it's 1/2 for
Oh,
what did I got
do?
the
Oh, I wrote
1/2
one.
and 2 1/4.
Okay, so we want. We've got 1/2 times 1/4. Oh, I'm on this. Yeah, on which one do you want?
Number one.
Number one.
It's
Okay.
going to be 1/2 times 1/2 times 1/4 times 1/4 times 1/4.
Did you get 1/2 for the double B's?
Double B's?
It's the same as the eights, isn't it? There's
Yeah.
1/4.
Yeah, it's going to be the
double
same as
big
the A's.
B's.
It's going to be 1/2 heterozygous B's.
This is asking for a big B, big B.
Oh, big B, big B.
Yeah, so I've got
Oops.
1/4.
Yeah, that's 1/4.
Okay. That's correct.
Yep. Yeah.
And then it asks for little c, which is 1/4.
1/4 again. Big D, 1/4. Little E, 1/2.
Big D, I've got 1/2.
Me too.
1/2.
Yeah.
Oh, yeah. Oops. 1/2.
And little e is 1/2.
Yeah.
I just want to
I don't feel like doing this in my head.
You don't feel like I could do it in my head.
You just multiply the bottom and then multiply the top.
Okay.
2 times 2 is 4. It's 4. 16. 32. No, 64.
256. Is
What?
that what you got on the bottom? Did you simplify?
Oh, dang it. This doesn't tell me. I can't. I got to get a real calculator out.
What is it telling you the decimal?
It just tells me the decimal. It doesn't
Oh.
switch it to fraction.
London.
Samsung hasn't got that capacity yet. Okay. 1/2.
Well, I got the decimal.
1/4. 1/2. 1/2.
The calculator.
1/128 is what I got. Holy
What
crap.
I got.
That's a big or little amount of chance of getting that.
You got 1/128 now? Or 0.0078125?
Yeah.
Cool. B.
Okay.
So cool calculators. Some sort of club or something?
Yeah.
The same ones. I got the 8th grade.
It's called the CCC Cool Calculator Club.
Yeah. Okay, so they're all little except for these, right?
Yeah, 1/4 times 1/4.
What portion of the offspring?
All the E's and D's are going to be 1/2.
1/256, right?
Yep.
Was it?
1/256.
Big B, little b.
1/2.
That's 1/2. Okay, little c, little, wait,
Low C, low
low
C.
c, little c.
1/4.
Little d.
Little d, little d. None. There's no chance of getting that.
0. Oh,
Tricky. 0.
portion.
Trying to trick us.
The nerve.
2 4
Okay, the other one. Can be
5 times
times
0.5 times 0.5 times 0.5 times 0.5. Oh my gosh. Maybe
Oh,
turn it
crap.
into a fraction again.
I got 1/32.
Oops.
I'll tell you.
1/32.
Yep.
I hope she watches all of our videos.
I know. I would not do that if I was a teacher.
Imagine like none of this. Yeah, million
Times like
Well, how many
many
students she has?
Yeah. All right, paired homologous chromosomes are sometimes called
paired bivalence, right? Isn't that a
You
paired?
just love that word.
I
It
do.
sounds
Well,
Is it?
I just learned it. I'm telling you.
Is it?
Well, I
Paired
thought,
homologous chromosomes are a tetrad.
Yeah.
But no, homologous chromosomes, you don't pair a homologous chromosome.
sure you do.
Not,
You do it in.
I think she's just messing with us.
She's not missing.
I don't think this is a,
Oh, it's a thing.
I mean, you make a synaptic cleft around, but paired homologous,
Here's the reading. Readings.
because this is a homologous chromosome. These are homologous to each other.
Yeah.
You don't pair them.
Whoa. Yeah, you do. Don't they kind of pair up
They
when
kind
they go
of do,
across the
yeah,
metaphase
I
point?
guess.
Mhm.
Okay, yeah. They're
So-
not like attached.
Kind of think they attach. Maybe a little bit.
We're
Go to the PowerPoint. It says
They do
Why I get
attach.
so confused.
With the synaptonemal complex.
synaptomema.
Right here.
Yeah, I'll do it. Is that what we call it?
That's what I called it.
Or what? What's another name for it? Metaphase plate.
But I mean,
No,
they're
that's
not
different.
called the synaptonemal complex. That's just what that thing is called that connects them.
Google. Wait, this isn't a quiz, is it?
I don't know.
After you complete.
No.
Okay, it's just a problem. So
Ah, here. Here's one. Here's something about bivalence.
Yes.
yes.
Synapses. Okay. Homologous chromosomes find each other and pair along their lengths, forming bivalence or tetrads.
There we go.
Oh, my goodness.
Right, bivalence
You
in all
feel
caps.
really good, don't you?
I feel awesome. How could I not?
How do you spell bivalence? You probably have it memorized
Oh, I do.
by now.
I do.
Just working the end of conversation.
Look at all
memory.
that hate, but
Or
it happened. I'm just ridiculed.
that's so funny. That's so funny.
Oh man. Yeah, awesome.
Thanks.
I didn't know that's how you did it. I always just erase those lines. You just put underline, huh?
Yeah.
Read the
Pretty fancy.
makes. Okay.
All right.
All right. Structure. John's got to get to class soon. So.
Pretty soon. This is a kinetochore. What's that called? Spindle or microtubule or something?
The structure makes the same thing.
It's a kinetochore something.
Oh yeah, there's a kinetochore spindles, or there's another name for them too.
Kinetochore microtubule
Yeah,
because they attach to their kinetochores.
yeah.
Structure consists of each of the microtubules.
Hmm.
Chromatic spindle is called a Kinetic? Oh, kinetochore.
Yeah,
Yeah.
I'm gonna pull up the PowerPoint. That's got a lot of info in it.
that's what it is. Because that's what those things attach to is the kinetochores, which is a protein found on the centromere.
So kinetochore spindle. Or
Which
What
is
do you-
No, no,
Kinetochore.
it's just asking what connects the centromere. This to the tubule is the kinetochore.
a diploid fruit fly has 4 pairs of chromosomes. Uh-oh. A fruit fly cell in prophase 1 has why? These questions.
4 pairs. So would it be 4 sister chromatids? What?
In
Four
prophase 1, it would be 4.
Prophase 1, right?
It would be 4. 4. 4, 2, and what is that?
the fruit fly
Has
cell
4.
in prophase has blank sister chromosomes.
4.
It's got 4 pairs of chromosomes. 4
Oh,
chromatids.
so it would have 8
8
Yeah,
sister
sister
chromatids.
chromatids.
yeah, because there's 4 pairs, meaning there's
Yeah.
So that means there's 8 chromosomes. So would it 4 of
8.
those be sisters?
Chromatids.
chromatids, the little ends of them.
I don't remember those at all. Okay. Because I thought that one Google picture said
This
that.
is like how
Remember?
many chromatids make up a chromosome? I must have asked that before. During prophase 1, the answer. The 2 homologous chromosomes come near each other. Because each chromosome consists of 2 chromatids, they are actually 4 chromatids aligned next to one another.
But 2
So-
sister chromatids. Right?
Sister chromatids make up a chromosome. Oh, goodness. Chromatids with sister
A
chromatids.
sister chromatid may also be said as 1/2 of the duplicated chromosome. A full set of sister chromatids. So it's just asking how many sister chromatids are there, right?
Yeah.
So 1 sister chromatid.
It never points to these 2.
I know that part.
That's my question.
Well,
Is,
it's like-
are those another set?
Or are these? Why would that?
And then this one shows those 2 are sister chromatids, but that's a different sister chromatid.
Wait, go back to that.
They never show both of them being chromatids. So it's like each chromosome has 2 chromatids, but when they're like that, there's 4 of them. Look, oh, you know what? Look at these. They're sisters.
So this is a chromatid, and this is a chromatid, and these are sister chromatids.
Oh, that's what we established.
Yeah, but it says how
But
many
you guys keep
sister.
saying ends and stuff. So if it's 1/2,
No, I'm pretty sure that a chromatid is just one. It's like the singular form of a chromosome.
no, this is a chromosome. What chromosome?
Yeah, this
And it
is.
shows the 2.
There's 2.
That's what I mean. Like a chromosome is a pair of 2 chromatids.
Yes.
Yeah. So then
Yeah,
the
and
homologous chromosomes would be like this, but
And so
Another one.
sister chromatid are these 2.
Okay. This is a chromosome. These are chromatids. These are sister chromatids.
Right.
Each half is a sister chromatid. There's only 2 sisters in a chromosome.
Right.
so if there's 4
There's not 4
pairs
sisters.
of chromosomes, there's 8 chromosomes. And if there's 2, that's there's 16.
Yes.
Say that again.
So if you have 4 pairs of chromosomes.
Okay.
There's 4 that contains the X.
Yep.
That means you've got 8 of the X's.
You have 4 pairs of them. Okay,
Yeah,
I just
so that's
read
there's
4. Okay, so
8
there's 4
chromosomes total.
So
Okay.
there's 8. So then if each one of those X's is a pair of sister chromosomes, there's 16.
Perfect. Yeah, that makes sense.
So that means there is
Okay, I think we got this now.
32, right? Because then it's going to make haploid. So it'd be like 4 for each. 4. Would it be 4 for each? Yeah, it'd be 4 for each. Because,
Be 8 for each.
no, because that's only for a diploid cell, a diploid fruit fly at the end of meiosis 2. That's going
Oh,
to create,
metaphase 2.
yeah, it's going to
Okay, yeah.
create haploid.
Mm-hmm.
So they're going to have 4.
Yeah.
4 times 4, so there's 6. Ooh. Yeah, because there's 16 at the end of phase 1. So those 16 are going to
Literally
split
copy
into
of it.
2.
Yeah, this is literally what's
It's
happening.
16.
There's 4 chromosomes split, or 4 pairs, but double this, whatever. So each of these has 4 chromosomes total in it now, once you get to telophase. So 4 chromosomes in each, okay?
Mm-hmm.
4 chromosomes still. 4 chromosomes still in each one. And then you get to metaphase. Each of them still has 4 chromosomes.
Yeah.
Each 4, okay.
No, they're going to
Well-
have 8 because we're in meiosis 1. So then the 8 is going to split in, or it's going to have 16, right? Because then 16 is going to split into 2 for 8 for each at the beginning of meiosis 2. The end of meiosis 2, there's going to be 4 haploid cells, so there's going to be 4 in each of the 4. So there's 16 at the beginning of metaphase 2.
But, so
Or no,
chromatids,
it's just
I've got
8.
8
That's
8
1
chromatids.
Yeah, 1 cell. Yeah,
That's what I got.
Yeah. You were
What?
you were right, but you were thinking multiplying.
Yeah,
by
yeah.
4.
Yeah.
Okay, I just did the extra work. Hey, Blake is a type of genetic cross that can be.
A leek is a type of insects that can be determined. The purple flowered pea plant has the pea. Big pea. Oh, where'd you go?
What are you talking about?
It's a type of genetic cross.
Would
So
it be
it's a
the
type of
F1?
genetic cross. Yeah.
Because the F1 is the secondary
Oh, yeah,
one, the
that's the
parental
secondary.
one. Wouldn't
Filial
that be
one?
right?
Filial.
Or it'd be like F2 or something. Yeah, it's filial or something.
Or parental. It's not the
I
parental.
don't think it's parental. I think it's actually filial.
Okay, monohybrid cross between 2 plants that differs in a single characteristic. Is that what it is?
Oh,
So, is a type of genetic cross that can determine if a purple flowered pea plant has the. Okay, so it's asking for just one.
Oh,
So it's a monohybrid.
then yeah, that'd be a
cross.
Okay.
monohybrid.
Okay.
good.
I always have that problem with.
You're just really
Stuff.
smart.
I just overcomplicate things. I just.
I mean,
Yeah.
it's help.
Is there anything push control B and then control U? Try it next
So
time.
when my
It'll be
fingers are
fun.
right here. I'm just kidding.
I don't know.
Too bad.
To me, it's
Yeah, I
faster.
know. I know. Probably true. Okay. We're still on 2.
Yeah, we're almost done.
Okay, good. A non-functional allele is sometimes called a
Non-functional.
Dud.
no.
A blank.
Sometimes it's just called a non-functional.
Is it recessive? That's not.
No.
Well,
Display.
but why would a dominant be only
Mass.
partially?
Oh no.
So it goes to middle. Genetic terminology. Let's try that.
Might be in her reading.
Polymorphic alleles. Okay, wait. A mutant allele. Rare mutations in genes can produce.
Oh no.
No. Can produce completely defective protein
If that's
product or
okay.
fails to produce a protein.
No.
No. No alleles.
Yeah.
Yeah.
Non-functional. There's also, wait, go back to that. real quick.
There's also silent, which
Yeah.
it doesn't.
That's not it.
Well, if it's functioning, well, if it's
Be
non-functional,
non-functional.
that means it doesn't work.
Non-functional.
A silent just says it's a mutation. Oh, it doesn't affect. Doesn't result
So I think this
in a
is,
defective protein.
yeah, but it's functional.
That doesn't give much information. This
This
mutation.
produces a completely defective protein. So that would be a null.
But that's not what this is asking.
Oh,
It's saying a non-functional allele is something, while a partially functional allele. Let's see if there's another slide. No. Okay. What is a non-functional allele called? Google or Bing?
didn't you say you get points or something?
Yeah, but I don't use them.
Oh,
Points?
you use B.
What
Who
do
gets points?
you call
Using
a
B.
non-
What?
No, not like that kind of.
No, I
points.
wish it was that. No, like I get points for like a card or something like that.
Like a gift card.
Man has a non-functional mutant allele.
I just type random stuff in all the time.
Yeah, like just think of a word and do.
Okay, Heidi, you just sit here typing.
See, not null allele.
That's the two normal function.
I think that's it.
Yeah. Well, a partially functional. What's the partially functional?
That is a good question.
Gain of function alleles. This mutation produces more protein products than normal, and the mutant protein works better than the wild type. It's not that.
I think it's even out there.
Yeah, I don't.
I don't even.
Okay, wild type. The most
No, that's
abundant
just the
allele.
normal.
Yeah, it's just the normal.
Partially functional allele is called a mutant allele.
You think?
But it could be fully functional, couldn't it?
Yeah, technically.
It just might be different.
Mm-hmm.
Well, that doesn't mean it's not functional.
Mutation for enzymes.
Epistatic.
It's a cool word.
Oh, interesting.
Oh. Just a second. Who said that? Epistasis.
Any
It was
clue,
in the slides.
Any clue?
No, I think I got the next question.
Oh, that's good.
Okay, what makes recessive?
Sometimes recessive. Maybe makes it partially dysfunction.
Okay, produce wild type. Gain of function mutations.
Tell me you need a loop, Joe.
It's in the. You just want me to go, don't you?
Huh?
You want me to,
That's
you want me
right.
to go, don't you?
No, I want you to miss your class. 42 pages. It's pretty impressive.
Hinkley, 221. It's kind of hard, isn't it?
Yeah, this isn't too easy.
Somewhat. Like if you left at 3, you'd be fine. I just don't know that we're gonna be done at 3.
I hope it's that we can get done. I need to go shopping the first one.
Yeah, you got a hot date.
Why does every question have at least 4?
It's really
5 questions.
Yeah, for
questions.
real. This said 4 questions, not 14.
I think they are going to be. We're gonna answer that one on our own later. Deal. Let's do some
Sounds
reading
good.
later.
Let's get the other answers.
Yeah. You said you found F,
I
John?
think it's the first one, the segregation.
Not even a question.
And the other one I think is independent assortment.
That makes sense.
Of course it is.
I said,
It's gonna be a long video for our teacher to watch.
uh-huh.
Hope she watches the whole thing.
Better. Yeah, you spelled independent.
They spelled it wrong.
You did it.
Autocorrect put it in that way. Just kidding.
All right.
Did
how
you
many
find
more
that
do
just
we
in the
got?
slides?
Well, if we go, it's a slide 2, law of segregation. Deals with a pair of homologous chromosomes that carry one allele for seed color trait, right? You go to dependent assortment, it doesn't say much, but if you go back to this one with the dependent assortment, you're dealing with 2 different things.
Yeah.
Bueno.
Clear any question? 3. Is it just 4? 5. Okay. Suppose an organism has the genotype F. Assuming that the TT alleles and R alleles and the YY alleles are stored independently. Diagram all possible homologous chromosome arrangements in this organism during metaphase 1. I forgot what all the names meant. Be sure to indicate which alleles in each pair of homologous.
That doesn't make any sense to me.
No.
That whole question doesn't make any sense to me. How can you draw
On the computer.
all that too? But how are you supposed to draw all the chromosomes and know where those alleles are? I don't get that at all.
Diagram all possible homologous chromosome arrangements.
So
Is it basically just all the Punnett square options for it?
Yeah, but then you have to draw the actual chromosomes, is what he's saying, isn't it?
That's what it
It's
sounds like. 'Cause
diagram
we
them all.
only have to indicate which alleles are found on each pair.
Of homologous chromosomes.
And this can't even be a Punnett square because it's only one organism.
Yeah, it's just going through the analysis.
Oh, that's not hard, actually. That's how you do it. So you got T, R, Y.
Try.
That's kind of symbolic there.
Yeah.
It's just trying to say something to us.
I think so, but autocorrect is trying to.
Oh, I see what you're doing there.
Is that all over with the T? T. Oh, no,
There's no little t's.
there's 2 big T's though.
Yeah,
It
so
could
it'd be,
either. So do we have to repeat everything basically?
yeah. Because you have to do one for each.
Yeah.
T.
Okay, there's one more.
many did you guys come up with?
So we've got 8.
Me too.
Oh, yeah. T. Oh, R, big Y.
It's really 4, right? Times 2. Like, they're the
Pretty
same.
much. Like, just because there's that second
Mhm.
big T.
So now what do we do?
Indicate all possible genotypes that can be produced.
Are you
Habit
eating
of-
your pen?
I have a bad habit of chewing on my pencils. When I'm thinking. So always. You should.
He's just hungry.
Which feels that too. I haven't had lunch yet.
Is that all we need for that? That looks diagrams.
I don't really understand,
What's in the diagram?
but that makes sense.
This is all, okay, so we've got all the possible homologous
That's all of them that could be
chromosome
found on each pair.
arrangements in this organism during metaphase 1.
I think that's good.
Be sure to indicate which alleles are found in each pair of homologous chromosomes. What?
Well, isn't each letter an allele?
That's what I thought.
So that's all it's saying.
So how do we indicate?
I don't know. Wouldn't it just be each one of those
Yeah. I'm
things
done
is
with
found
that.
on a homologous chromosome?
I think that explains it. If she gives some more direction, maybe on that one. I don't know on that one. I think I'm just gonna put that one.
This is a long video.
How long is it?
Multi-hour study session. We got in here at like what, 1:30 or something.
A little over now. Indicate all possible genotypes that are produced in this organism at the conclusion of meiosis.
Same thing.
Sure. I don't
What do
know why
these all
they would
mean?
change.
Like, you know.
Well, I thought-
Oh, so this one I think we can just put them all one time.
we already did them one time.
I mean, like, I did them, like, you do beat each of these 2 times.
Because it's just
basically,
half of it, is what you're saying?
Yeah.
Sure. I don't know any other way to do it, so I think
Yeah, I really don't know what that's. How many sister chromatids are found?
What?
Okay, I think we're doing this wrong.
I think so too. Wait,
We do have to diagram it. So we have to do some drawings.
links.
we don't have to draw the whole semester. So we can't even do this one until we print it.
I say we call it good for now and do it on our own, and then if we need to, because this isn't due till Saturday,
Right. We can meet up again or
we can maybe meet up again or just talk to each other.
When would you guys be able to meet up again?
something. I don't know.
I feel like
Tomorrow there might be a chance of meeting up.
this number 3 just seems confusing.
Yeah, tomorrow I can meet anybody if we want to.
But these are alleles, right? So aren't different alleles, like is it one chromosome for one thing?
What I think they want us to
Or is it like there's 4 or 3 different chromosomes? And like one chromosome is got the TT allele, one another chromosome has the RR allele. That's why that is. That what it means?
I think that's what it is. It's like one is going to have
The
the
TT.
TT, one will have the. You can go TT, TT, and then R, little r. I don't know how to do that. Because each of these has two things in it. Like this will have T and T, or will it have just a T?
Well, so you can find different alleles and different chromosomes. Like who's to say it's not all in one chromosome?
It's because they assort independently, so it has to be separate chromosomes.
Oh,
Because if it was,
yeah.
if they assorted dependently, they'd all be the same
Same.
chromosome.
So
Because these did a crossover earlier. And once they did the crossover, it gave this maybe a R, like a big R, little r, or something like that. So this will have this could have the big R, little r, but it doesn't give us a secondary thing. So it would have got a big T and a big T from, like one big T from this one.
Hey guys, we're gonna have to kick you out because we gotta reset this room, so we're gonna sell your tables. Sorry.
Let's turn like in this one now. And so you think we can submit this thing now? I think so too. I don't see how you're watching or if you guys did. We did the main part, and I don't know if we're even supposed to.